Good morning, viewer. Bienvenue here. Here's the most amazing news for our viewers. Yeah, MIT researchers have engineered a water-soluble histidine kinase, a bacterial enzyme that could lead to a new class of antibiotics aimed at addressing the growing issue of antibiotic resistance. This development also paves the way for using similar techniques to mitigate environmental issues like greenhouse gas emission. A bacterial enzyme called histidine kinase is a promising target for new classes of antibiotics. However, it has been difficult to develop drugs that target this enzyme because it is a hydrophobic protein that loses its structure once removed from its normal location in the cell membrane. Now, an MIT-led team has found a way to make the enzyme water soluble, which could make it possible to rapidly screen potential drugs that might interfere with its function. The researchers created their new version of histidine kinase by replacing four specific hydrophobic amino acids with three hydrophilic ones. Yeah, even after this significant shifts, they found that the water soluble version of the enzyme retained its natural function. No existing antibiotics target histidine kinase, so drugs that disrupt these functions could represent a new class of antibiotics. Such drugs. Such drug candidates are badly needed to combat the growing problem of antibiotic resistance. Each year, more than 1 million people die from antibiotic resistance infection, saying Shuangguang Zhang, a principal research scientist in the MIT Media Lab and one of the senior authors of the new study. This protein is a good target because it is unique to bacteria and humans don't have it. Ping Zhu and Fei Tao, both professors at Shanghai Jiao Tong University, are also senior authors of the paper, which was published on June 10 in Nature Communications. Mingangka Li, a graduate student at Shanghai Jiao Tong University and a former visiting student at MIT, is the lead author of the paper. Many of the proteins that perform critical cell functions are embedded in the cell membrane. The segments of these proteins that span the membrane are hydrophobic, which allows them to associate with the lipids that make up the membrane. However, once removed from the membrane, these proteins tend to lose their structure, which makes it difficult to study them or to screen for drugs that might interfere with them. In 2018, Zhang and his colleagues devised a simple way to convert these proteins into water-soluble versions, which maintain their structures in water. Their technique is known as the QTY code. For the letters that represent the hydrophilic amino acid that became incorporated into the proteins. Leucine L becomes glutamine Q, isoleucine I and valine V become threonine T and phenylalanine, that is called the F, becomes triosine Y. Since then, the researchers have demonstrated these techniques on a variety of hydrophobic proteins, including antibiotics, cytokine receptors, and transporters. Those transporters include a protein that cancer cells use to pump chemotherapy drugs out of the cells, as well as transporters that brain cells use to move dopamine and serotonin into on out of cells. In the new study, the team set out to demonstrate for the first time that the QTY code could be used to create water-soluble enzymes that retain their enzymatic function. The research team chose to focus on histidine kinase in part because of its potential as an antibiotic target. Currently, most antibiotics work by damaging bacterial cells walls or interfering with the synthesis of ribosomes, the cell organelles that manufacture proteins. None of them target histidine kinase, an important bacterial protein that regulates processes such as antibiotic resistance and cell-to-cell communication. Benda's exclusive report.